leading now this university as a dean, research and development. So I wish that Rohit can come and take this talk.
Good morning, everyone. So I thank Madam for a very nice introduction, brief introduction, and I thank Dr. Kumar and Dr. Deepa for inviting me for this presentation and having an interaction with you all. What Madam just said is very correct. There are very few taxonomists left, and the interest of taxonomists have reduced over time. But I'll like to give 2 examples before I start my talk. There is Professor Kumarik. He's a cyanobacterial taxonomist. He's 90 years old, and still from all over the world, people send their samples to identify to him. He has a microscope at home, and he's still working. Another is Professor Stevenson. Just last week, I heard his talk, and he was giving his talk from his home, and on the background, he has his microscope and culture plates being there. And he's around 80 years old. And he is told that when he was active, so he used to culture the plates, 50 culture, like moist chamber culturing of myxomycetes is required. So he used to culture around 50 cultures per day, but his now efficiency is reduced. Still he is able to do 15 to 20 cultures in a per day basis. So still he is active. So it's a very good field. People should try to have any kind of thing and try to go into the field. To start with the present species concept, in the initial part of the talk, you may like that it's not a fungal talk, but I like to try to bring you a kind of basic outline of what species concept is. This species concept, the term, is very simple, and when we try to identify, okay, what is a species, we think yes, we will be able to define this species. We are listening this term since from the 10th class, 12th class, in our graduations and PG, everywhere. But if you say that it is still no satisfactory definition of what constitutes species has been proposed. That was Rowensky said earlier. And first it was Darwin. No one definition has yet been satisfied all the naturalists. Why I am trying to give this history is that because the earlier all the species definition for fungi was not for the fungi, it was for the microorganism, that is plant and the animal. So whatever earlier species definition we were able to know was based on the plant and animal, because at that time microscope was also not there, they were not observing the microorganisms. So whatever species definition they were giving, or the criteria for the species, there are 2 things, concept and a criteria. Concept is overall concept, and to define that concept, what criteria we are using to reach to that concept. Okay? So these 2 things, words I will be using quite often. So there were a lot of ambiguity on the definition of the species concept. But still, so many years of taxonomy has been going on, but still species is still the basic unit. We have been able to identify now, strain differentiation is quite easy, very easy, we are able to differentiate between the strains, but still species remains in the hierarchy of taxonomy, the basic unit. Okay? And why? It is very easy. In biology, a species is one of the basic units of biological classification, but it's earlier it was capable of interbreeding and producing a fertile offspring. This definition was based on the animal taxonomy. Okay, so many of the earlier criteria that was used was based on the okay, this thing. So now it is audible? Yeah.
Yeah, please proceed.
Yeah. So after that, it was most of the people have defined different species concepts. So there are different 26 species concepts that have been given by different taxonomists. like biological species concept, type species concept, and ecological species concept, and so on and so forth. So there are different species concepts that were given by different taxonomists. Like for later on it was evolutionary species concept that was given, kind of thing. Basically the common ones are these are the four, that is the typological, morphological, biological, ecological, and phylogenetic. The typological species concept is the emphasis on the type material, that is type material deposited in the culture collection. To give a brief about type, that means the type specimen or the culture on the basis of which the new species has been designated or defined. So that is type. Then morphological, that is the based on the morphological divergence. Biological, that is based on the interstellarity concept. Based on the ecological, that is the ecological concept. Ecological, I'll later on give some of the examples of the ecological diversity, why it is going. And now it is the phylogenetic, and definitely combined at present, what is followed is the polyphasic species concept. Polyphasic means combining all the things. Okay, so that is why we are having a polyphasic species concept, because we are not using a single criteria for the kind of thing. We are using all the criteria holistically for the defining a new species. So morphological species concept. There were several examples of morphological species concept. Fungi has also faced a lot of problem because there were dual name for a single species, Aspergillus separate, Emaricilla separate, and those kinds of things was a lot of problems were there. Glomerula, fusarium, and all these problems were, lot of problems were there in fungi also. And some of the examples I like to brief you about this hermit crab, that is where you can see this is a kind of polykey tube worm, where it has one form is this and one form is this. So it is the two life stages of a single organism. Like if you imagine a kind of any plant, you imagine it has two kinds of stages. It has a vegetative stage. Just imagine a coriander. You see when you coriander, it is in a vegetative stage, it looks quite differently. When it is in the flowering stage, it looks quite differently. You see aloe vera, it looks quite different in the vegetative state, it looks quite different in the flowering stage. So earlier when the scientists used to observe these kind of things, so they used to think these are the two species. But the moment DNA sequencing and all those things come into frame, or the complete life cycle were observed, then people come to know these are not the two species, these are the single species, and they were giving two names to the single species. So these kinds of problem earlier a lot of people have faced because of this molecular things also, as well as non-observance of the complete life cycle. Similarly, reproductive isolation. So this is reproductive isolation. We cannot, it's difficult to use it in fungi, but it was quite popular as compared as animal is concerned. So mating experiments for all organisms is quite difficult. If you include this criteria as a species concept, it will be difficult. If I want to define a new species of fungi, then I have to check its mating with all the 300 species of Aspergillus on any of the fungi. It is quite difficult at present. And it is quite difficult in animals also. Although they tried, like there are two African species are there, they tried to mate it, but their offspring was not viable, it was not fertile. So that's why it was found, they have tried to do some of the experiments. Like if the reproductive isolation is there, you imagine whether in cow or in kind of different breeds of dogs, whether this reproductive isolation criteria fits. No, it cannot fit. Getting point? So this is not a good criteria to define it. Then hybrid species. There is lot of hybrid species one level, but since one of the species is we do not have a live specimen, we do not know whether they mated, whether they have the offspring, or whether they really are two different species or not. It's based on only the fossil things. So this cannot be proved into this animal or the human thing also. So the reproductive isolation or non-fertility can be a genetic also, can be geographic also, can be behavioral also. So many of the criteria cannot be tested in the species concept. Okay. So these are fairly common misconceptions that proceed to misunderstanding of the biological species concept, which makes species distinction based on the fertility. So there is no criteria that says that if two populations can interbreed, they are the same species, and there is no criteria that says that if two species can't interbreed, they are the same species. This was regarded as the barcode of the fungi. So previously, even a plant pathogenic species found on a new host would have been considered as a novel species because of the physiological specialization, and there were several of the criteria like that. F species used to make several of them. But however, phylogenetic studies have changed the concept. So there were around, I think, 13 or 14 trichoderma species which were what we call as cryptic species. Okay, when you are able to identify biomorphologically only 2 species, you have 5 strains, but you are identifying the through morphology, you are able to identify that these 5 strains belong to only 2 species. Okay, the moment you do the ITS sequencing, you found that these are not 2 species, these are 5 species. Okay, so these 3 species that new have appeared in the sequencing, these are called as cryptic species or hidden species, which you were not able to resolve or identify based on the morphology. Okay, so what is the species delineation at present? It is the morphological, physiological, interstellarity. Also, I'll explain why I have mentioned it, although it is not a regular criteria, but it is sometimes used. I'll explain how. Host specificity still is a criteria, and a phylogenetic, but not a single. It is all holistic. By one thing you cannot do it, but holistically you can do it. Here I like to define 2 definitions more. One is identification and one is characterization. When I say identification, I want to identify a strain, that's all. So what means I will follow? Either I will follow phylogeny, like I will do the ITS sequencing, blast in the gene bank, and then see what is the identity. Or I will do the morphology, and based on the morphology I will identify what is the species. Okay, these are the 2 identification methods we are following. For yeast, still API you can do it, but for mycelial fungi, these 2 we are following. If I want to characterize a fungi, when I say characterization, these are already known strains. I just wanted to identify it, done either sequencing or morphology. But when I want to characterize a strain, that means I have found a new strain and I want to completely characterize. Then I will do I'll generate all the whole data. I'll generate the morphological data. I'll generate the even physiological data, at what temperature, what is the optimum pH, what is the carbon source, nitrogen source, everything I'll try to characterize. I'll generate the whole data. Why? Because that strain or that species, I am making it known to the world for the first time. So the maximum description you will be needed for that particular strain or the new species that you are proposing. So that's why whole thing is due. So this is characterization, and identification you can do by sequencing or by morphology. How to proceed for a novel fungi? Before I'll go, I'll just briefly explain you, and then we'll go to the further talk. So previously, since the sequencing was quite costly, so when I was doing my PhD, at that time what we used to follow was, I had around 50 strains, so I'll identify through morphologically, those I can identify through the literature. And at that time literature was also good in all the laboratories, previous laboratories. So we used to follow that. I'll follow 50 strains, I'll do the morphologically. Based on morphology, I identify around 40 strains I could identify up to species level. 10 strains are left which I cannot identify. I have doubt, I have confusion, and they are identified up to the genus level. So what I'll do the sequencing of those 10 cultures. It was costly at that time, but now with over time the sequencing cost has reduced. So the working of the people have also reversed. So they first do the sequencing and then see what are the novel things, and those which are required to be those which are not identified through the gene bank database. At that time database was also obsolete, but now the database is also quite rich. So now people are able to do the sequencing. After 50 strains sequencing, you come to know that okay, 5 strains are there which are probably novel, which is showing less similarity in the database. Those you can completely characterize. Now generate the morphological data, physiological data, and all those things. So what I follow is, I do the sequencing. Mycelial form, for mycelial form, ITS is good because ITS is the barcode as well as its database is good. For yeast strains, if you have a yeast, you do the LSU region sequencing first because its LSU database is better. When ITS has to be done for yeast, I'll let you know in the later stage. but the primary region for the yeast will be LSU. Then you do the blast and search based on the similarity. If it is low similarity, then you do the morphological characterization, description, physiological studies, that is pH, temperature, media, etc., whatever generation. Why I am saying these criteria are there, I'll just explain. Sequencing of other regions, let LSU, SSU, other protein coding genes, and then you construct the phylogenetic tree. Okay. Now, why I am saying physiological data is important, because like suppose we have studied in Lunar Lake, we have isolated around 108 strains of fungi from Lunar Lake, and there we found that many of the strains were alkaliphilic. They were from the alkaline environment. So their pH was quite higher, around 10. And after below 7 or 6, they were not able to grow. So that's why any strain that you are isolating from any plant, any rhizosphere, any place, you try to test its pH and all those things. So that is very important, significant for your studies. And then the phylogenetic analysis. So this is the overall things that one has to do. Like you have to do DNA studies of any of the fungi if you are isolating from any rhizosphere, any biofertilizer oriented strain, or any plant pathogenic fungi. Morphology you have to do. Physiology also you have to do. Then extrolytes in many strains like Aspergillus, Penicillium, Fusarium, Colitrichum, which are complex, which are having more than 300 species. There you may also have even the multi-region MLSA, multi-locus sequence typing, sequence typing will also not able to resolve you. So that's why at that time you may have to do the extrolyte profiling. That is different strain produce different extrolyte in Aspergillus, Penicillium, Fusarium, Colitrichum, these things. So at that level you have to do. That means you have to generate morphological data, physiological, biochemical, your DNA ITS region. Then you have to see which region, multiple regions are required, protein coding genes, calmonidin or beta tubulin or whatever, and then finally extrolyte also for the complex genera. Okay. Ecology. Ecology, why I am saying ecology is very important. Ecology is important like for in agriculture, like if you are working on some mushrooms. If anybody comes and collects a mushroom from the forest and tells me, okay, this mushroom I have collected from soil, and he says that this is pleurotus, I'll say no. It cannot be a pleurotus because pleurotus only grows on wood. If anybody collects a mushroom and comes to me and says this I have collected from a wood and this is agaricus, I'll not believe it because agaricus only grows in soil. So that at this kind of stages, these ecological aspects is very important. Like Ganoderma, it grows on wood. Okay, but many times what happens when the roots are underground, it is coming from the soil, it appears that it is growing on the soil. So some of the students may collect it and bring it, they can say that okay, this I have collected from the soil. No, it is pathogenic to a tree, plant pathogen to a forest tree, so that is why it only grows on the tree. So in this way ecology is very important. So that's why while working on some of the fungi, this is very important. So definitely genetics, mating tests are also sometimes important. There is 1 or 2 studies that I will show wherein even after phylogeny they were not able to resolve, but mating experiments resolved that okay, these are the 2 strains are different because they were mated and they produced the fertile offspring. So that's why they were able to define it. Genomics, definitely genomics now has come into play, like your whole genome sequences has come into, although this criteria as far like in prokaryotes, the IGSCM society has made it compulsory for the genome sequence of the bacterial cultures. But in fungi, for taxonomy, still it is not able to define it because of some of the quality parameters and all those. Its data generate is quite huge, 5 GB to 10 GB, so analysis is quite difficult. And there are not several projects that is running out. In prokaryotes, what they have done is there is a international project that is running. You deposit a culture there, they will do the whole genome sequence for you. But in fungi, because of all these factors, still they have not made it mandatory for this whole genome for a taxonomic purpose. Okay. So starting from the morphological characterization, definitely the morphological characteristics that you are defining it will be based on several of the morphological features that different organisms they make it. Okay, they may be the zoospores, spongiospores, conidia, based on which genus or which species you are studying on. Okay, you are either studying, if you are studying phytophthora kind of thing, they are not, although not true fungi, but if you are studying, then zoospores will be produced by them. Then some of the fungi you are studying, maybe mushrooms you are studying, then basilospores will be produced by them. So in this way, different thing will be produced by different organisms. Okay, like these arthrospores, arthroderma kind of fungi, these are plant pathogenic, but these are animal human pathogenic. But these may produce different organisms will produce different structures. So based on that thing you have to see. So for this you have to sit on the microscope. You have to observe it. You have to see what structures are produced by it. So based on that you have to define what genus you are able to reach. As a taxonomist, first try to reach up to the genus level. See on daily basis one new genus you should be able to see at the morphological level up to the genus level. If you are able to identify up to a genus level, slowly, at least 50-60 fungi you will be able to identify very easily in a year if you are able to sit in the microscope. First try to sit in the microscope and then observe it. Just try to make any plant pathogenic fungi, any soil organism that you find, just make a slide, good slide and do it. For some of the things you may need to do slide culture technique, because in slide culture technique you will get good attachment, because until unless you do slide culture you will not be observing as a good attachment to the organisms. So different other structures, like if it is an ascomycota, then ascus and ascocarp you have to observe it. Then if it is a basidospores, then bastomycota, then zygospores, then zygomycota kind of thing. And try to follow the new classification thing that has been followed by Hibbett et al. And later on 2000. So where they have divided based on the SSU and ITS and all those regions, they have made a new classification instead of having that thing. Okay. So I will not go into that segment, that is why Deuteromycetes was removed and now which are different, because it is having the same story as I just told for the other organisms. It was having two same species having two names. So that is why you have to see a kind of thing like this, the basidospores from the Rasula species that we observe. So based on that you can have it. So definitely different. This is Chetomium that that produced good. This was the new species that we published. Then Clistothecium. So different structures you have to observe it under the microscope, and then like this structure. So this structure we observed that it was very difficult to identify what kind of structure is there. But when we cut this section, then we could observe that individual pores there were having the pycnidiums were there. It is very good in kind of wood rotting fungi, like in kind of there is a wood rotting fungi, Daldinia. So Daldinia also, if you cut a TS kind of sections, it shows very good pycnidiums coming on the pores on the surface of those things. Morphology in petri dish is very important. When you are culturing and when you are studying in a there are two kinds of organisms, culturable fungi and non-culturable fungi, or uncultured, not culturable, uncultured fungi, because we have not been able to culture them till now. Okay, so one is cultural fungi, which we have been able to identify its media and we have been brought it into the petri dish, but there are millions of organisms and lakhs of organisms there in the soil still that we have not been able to culture it. That's why we found in lakhs of OTUs in the metagenomic studies. So those which we are able to bring into the culture, they are very specific to nutrients, whether it is carbon source, whether it is nitrogen source, whether it is any other source. So you have to know your culture, you have to know your organism, just like your small baby. You know from the birth how you have isolated, how it was looking at the 3rd day, how it is looking on the 7th day, how it is looking on the 30th day, like that. One strain that we isolated, its optimum growth was 90 days. So it used to reach 3 mm or 4 mm or 5 mm, 1 centimeter, like 10 mm in 90 days it used to reach 10 mm. So it was very difficult for us to preserve it, because we need an optimum inoculum, because we preserve for preservation we need around 5, 6 or 10 discs for preservation. So to have 10 discs we have to inoculate 10 plates, then we were able to get in 90 days. So you have to know what kind of organisms you are handling, what is its optimum growth in 7th day, 10th day, whatever kind of thing. So you can see here, if you see this organism, you see here this one and you see, the morphology is quite different, but these are at the different temperature, 15, 18, 19, 20. It's behaving differently in different temperature, and the same you will observe in media. So you have to record what is your isolating media. If you are isolating in PDA, try to maintain in PDA. The moment you change in MEA, its morphology will change. You will see that, oh, my culture is behaving differently. It is maybe contaminated. But no, because of the change in the media, you have been able to. Number one. Number two, if you're isolating from some kind of, what I should say, extreme environment, some kind of environment which is not normal, so first try to have some of the isolating media from that also. Okay? Like if you're, somebody is isolating from kind of Konkan area, so there are very good studies going on in Konkan areas where people are isolating what I should say, high saline organisms from the rhizosphere, and then from the endophytic rice, and they are using it for the increasing the productivity or protecting from the kind of thing. So because they are indigenous strains, but for that they have to maintain high salinity. Okay, although the salinity of the ocean has reduced from 4% to 3.5% thanks to the global warming and the climate change, but still 3.5 salinity is quite a high for the fungi that has been isolated from the marine environment. Okay, so try to know what is the, and always try, I'm deviating from my present talk, but I'll just try to suggest you that try to have a pilot experiment. Whenever you are isolating, whether you using antibiotic, try to have 2-3 combinations of antibiotic, tetracycline, chlorphenicol, amoxicillin, 1:1, 2:2, and see which one is behaving better. Try to have different pH, try to have different media, and see which one is there. For many of my studies, I have used around 35 media for my primary isolation. Based on that, I found that, okay, these are the 10 media that are good, producing different diversity of fungi. Some will produce repeat kind of thing. Some will produce, okay, these are the organisms, but some will be producing maximum diversity. So since my kind of requirement was to have maximum isolations from the soil, so I used to first isolate in 35 media, reduce it to 10, and then those 10 I used for my studies. If I would have not done the pilot studies, I would be using 35 for all my studies, and out of that 25 are of no use for me. So that's why try to do a pilot experiment and then standardize it, and that will justify your study also. Because any external or any expert will ask you, why did you use this antibiotic? You can say, I used 4 combinations in my primary study or pilot study, and based on my pilot experiments, I reached to this conclusion. Okay? So try to think of pilot experiments whenever you're starting on a new niche or a new field. So this is slide just to make, in case of Aspergillus and Penicillium, there are specific media for isolation. It is not that you can show your petri dish in any media. There are specific media that are being kind of identified by the International Commission. So only these media you can use it for Aspergillus, these media for the Penicillium. So then only you can compare the growth, your morphology and everything, because spores also changes its size. There are some research papers where even spores have been observed to change their sizes based on different media. So try to see which media it has been grown. Earlier researchers have grown. Okay. So definitely the same media, then morphology studies you have to do, the complete data you have to procure. At least try to have at least 20 to 50 micro observations and micro measurements you should make based on the now many persons do not use camera lucida. Most of the software, they have the softwares for the microscopic pictures. So but try to take at least minimum 20 to 50 observations per thing, take the average and then report it. So this is the mating experiment, although mating experiments practically it is not very correct, because it is impossibly unwidely to perform mating experiments on all the millions of fungi that have been named species in the past 500 years. If Aspergillus has 300 or 400 species, how can I test all I do not have the access to all the culture collections. I do not have access to all the strains. So practically it is not possible to use. But sometimes at present also, in 2020, 2023 also I have seen some of the mating experiments that try to resolve it, some of the experiments where this Aspergillus fellis and Aspergillus udagavi, this paper is very nice, you can study. They have tried to find that yes, these two species are having two different species, because they could not mate it, so that's why they were found to be a different species, and then based on that Aspergillus udagavi was described as a new species. So there are still new species that have been described as a last resort, even after phylogenetic resolution. If it is not able to resolve, then okay. Then if you have some yeast there, so yeast has a different criteria. Okay, yeast, you know, as these are still fungi, but these are fungi as a unicellular organisms. So for yeast, which maximum belongs to the Ascomycota, some may also belong to the Basidiomycota, may form these three structures. Either they will form unicellular structures or they will form pseudohypha. These are pseudohypha or candida, so they will form as a pseudohypha. These are the true hypha. With there you have a branch. This is formed by the mycelial fungi, but pseudohypha is formed by the yeast. So it is basically elongated cells and branch cells, but it looks like a hypha. So that is why it is called as a pseudohypha. Do not confuse with that. So these are some of the photographs of this thing, like candida, and these are some of the pseudohypha, that is elongated branches, but these are not the true kind of thing. So this dimorphism, so for yeast you have to observe whether they are forming a single forms or they are forming dimorphic kind of form, and which kind of structures they are forming. Like Saccharomyces, which is commonly used in bread making, other things, you will not find pseudohypha in that. They do not form. So they will only bud. You will see budding stage or the single unicellular stage. So based on that thing you have to observe. Like many of the Clavispora yeast strains are now agriculturally very important. They are being used as a biocontrol agent for most of the pathogenic fungi. So even do not, if you are anybody working on a biocontrol aspect, try to also focus on yeast, because many of the yeast are producing antibiotic and they are also producing as an antifungal compounds. So they may have two forms. Yeast may have two forms. When you see, I am not saying two stages. Either these are, you can say two stages of the same fungi, or you can say in two forms. Okay, but they have a two. So histoplasma and all these examples, although are a plant animal pathogenic, but these both stages you can observe that these are there. So one of the important thing that you can identify for API kit is also good, even for some of the plant pathogenic fungi, like, or the agricultural important fungi like Aspergillus, Penicillium. Their database is quite good. So you can also use API kit for them. In API, what is happening is it is nothing but sugar tests. They have miniaturized this sugar test. So it has around 20 or 20 these strips are there. So if you see, this one is 0, that is control. You can see the red lines there. So red line has a purpose in there. So if you see red line, here you can see red line, and suppose glycogen source is there. There is no growth. That means it did not utilize it. Here it is in glucose, there is a growth, but still red line is little bit observed. Okay, so that means it is growing it, but it is optimal growth is there. Here you can see that complete this here and here complete red line has been gone. So that means here it is growing very nicely. It is utilizing the source very nicely. So API kit works in this way. So there you have to have a kind of a standard has to be maintained, like standard culture has to be made with a kind of thing, and then standard culture with equal kind of, what I should say, number of cells, of each cells. There you have to inoculate here like 5 microliter or 10 microliter, and then it will grow, and based on that plus minus, the software will give you the result that what is there. And based on that also you can see, or based on the plus minus thing also you can note down the results based on that. But software gives the genus level identification. I have seen that it gives nicely. Okay, and now an additional aspect of this is very nice, like biolog plates have come. Biolog plates are also good if you are working on the ecological aspect of the soil microbes of soil fungi. So biologs are also good eco plates. Their triplicates of these sources are there, so automatically you will get results in triplicates. Okay, so that is also good. So it is for yeast and fungi. API 20C, this API kits are used. So you have to make, first the important aspect is a standard. So you have to make a standard that the individual thing should have the equal number of cells. Then mushrooms. So if you are working on mushrooms, so based on that you can have a different thing, like basic different parts of the mushrooms. I'll not go into that. But what are the characters one has to do? Here the characters changes. In mycelial fungi, you see it was different criteria. In yeast, it was different criteria. But if you are working on mushrooms, you will have to observe different criteria. Here you will not observe the zoospores, you will not observe the mycelial breath, you will not observe the spores spores still you will observe. Mycelial width also you will observe, but many of the things that you observe there, you will not observe here. Like culture morphology, you will not observe here. You will have to observe the annulus, stipe, habitat, these things, different criteria it will be there. Okay. Here the color atlas makes an important thing. Here I am using the Rayner's color chart. So it's available. You can check on the online. So it's color, because you have to give a standard color. Because gray can be grayish also, light grayish also, dark grayish also. For me, which is gray, for you it may be light grayish. So it is quite ambiguous. So you have to give a standard chart by which you are following. So every chart has a number, like it is 6. And if you are following this one, so it will be 6 and 4. Sorry, it is F, D. I think B and D, something like that. Yeah, D. So it will be 6 and D, you have to learn. So if you are mentioning this one, it will be F and this 6 and F. So in this way, you have to give a number for that thing. So which gray you are mentioning, which yellow you are saying, which orange you are saying, because it will be different for different per person. So definitely there are different morphology. There may be some mushrooms which are gill fungi, some mushrooms which are having teeth, some mushrooms which are having pores, some mushrooms which are having these kind of pores, zigzag kind of things. So different mushroom will have different things. I'll give you some. These are some of the criteria that you observe, like pileus, size, surface. All these you have to observe. Whether it is central, whether it is eccentric on one side, lateral. This is mostly oyster. This is sessile, like Ganoderma kind of thing. This is Canthurellus kind of thing. This is Agaricus. So in this way you have to observe. So this kind of data sheet helps you in the field. If you are collecting any mushroom or working on a kind of thing, so you need to observe. Like if I am going in the field, I found this mushroom, I'll just tick mark it. I'll not memorize, okay, whether it is squamosous, smooth or whatever, I just have to tick it in my front. So you can print it from that thing. It's Lodge et al. 2000 paper. So you can get the sheet from there. Or you can write to me, I'll just forward you the sheet. So these are different pileal shape, different pileus margin, different gill attachment, different gills and lamella, different gill margin. Here you can see that either it can be even, it can be serrate, it can be wavy, it can be eroded, it can be like that. Then it can be whether it is attaching to the margin, whether it is how it is, here. Whether it is free, it is not coming to the stipe. Here it is attaching to this stipe. Here it is directly attaching. Here it is making an elongated up to this type. Here it is becoming decurrent. So different gill attachments. So all these criteria one has to observe. Similarly, stipe, different structure. Normally you find this volva either in, it can be a manetta also, it can be volvula also. How you will differentiate? So based on the gills, volvula you won't find in the volvula, but in a manetta you will find that thing. So based on that thing you have, and one is edible, one is poisonous. So it's very important criteria that one has to follow. Spore structures, definitely microscopic thing has to done. Different structure has different thing. So mostly these are formed by asterus kind of thing. These are mostly by Canthurellus kind of this kind of thing. So this is asterus, a very nice mushroom. So even when you cut a section, you find this kind of thing. So middle is lamella. On one side you will find this fertile layer. Here cystidia are there. That is a non-fertile basidia, and these are the fertile basidia where 4 basidia are there, and these are different structures of middle thing, whether it is straight, whether it is decurrent, whether it is inverging, all those characters you observed in the mushroom. So you can observe that all the three, like mycelial fungi, yeast and mushroom, have different 3 criteria. So I have tried to give you an outline for all the 3 things so that the kind of fungi you are working, you can follow that thing. Okay. Molecular characterization, you are already doing the practicals, so maybe practical theory must have been taken up. So ITS 1 and ITS 2 is the region that is more important. Which PCR you have done today? It is for the ITS region? ITS? Yeah. So you must have taken this ITS 1 primer and ITS 4 primer, that is the forwards and reverse. So it's in my observation, like when I was in NCMR, we were handling around 25,000 cultures of fungi. and we had sequenced around 15, when I was there till that time 15,000 cultures were sequenced. And based on my observation, these region ranges from 300 to 700. It's a quite wide range, but the average is 500 to 600. So most of the regions that you'll find is 500 to 700. But some of the east that we found was even 300 of the base pair region was there for ITS and ITS2. Saccharomyces it was, I remember. And one of the fungi that we found, it was 700 ITS was there. Okay, and sometimes one of 5,000, you will find that your ITS1 primer is not attaching, even after 2 and 3 thrice. Then you try to see that genus morphologically, try to see and see whether there is some mutation or some deviation in the ITS primer, because we call it universal, but there may be some changes in some strains where the ITS1 primer is not attaching. Then you have to design your own primer. But this we have happened only 1 or twice in the whole of my handling. Otherwise, these two are very good universal primers. It attaches very nicely. So these 2 things, and when you will be doing alignment, you will see that this 5.8S region is common in all. It is conserved. So in fact, it helps very much. Like all the sequences are aligned in 5 to 3 dash region, and one is reverse oriented. Then it helps to see whether your sequence is reverse oriented or not. So you will see that this has to be very equally aligned. So in this way. So for D1, D2 region that I was telling for LSU, this D1, D2, we do not sequence the complete 28S, that is more than 1400 base pair, but we use only D1, D2 region of the LSU region, that is around 600 to 700 base pair, because that is hyper diverse. So for complete MLSA, you may have to do multiple assay regions, where apart from these regions, common regions, you may also have to do beta tubulin, RNA polymerases, calmodulin. So you will say which region we used to use for which fungi, or all the regions for all the fungi. No, it is different. That is why I am saying different criteria used for different genus. Okay, all the criteria is not used for all the fungi. So you have to see the literature. So based on the literature, I'll just skip and then come back. So based on the literature, you will see what is the criteria. Like in Ketomim, it is morphology, ITS, LSU, beta tubulin. In case of Aspergillus, morphology, ITS, beta tubulin, calmodulin, actin, TF, RPB2, and extralyte profiling. So different criteria is being used to achieve the same species concept. Okay, so you have to see the literature. Based on the literature, you decide which region to use. When I was around 4 or 5 years ago, for Arthenium, only morphological and ITS was used, but now MLSA has also come for Arthenium. Okay. So this was the kind of extralyte that I was telling. The different strain will have a different extralyte. I'll just move little forward for because it's almost 1 hour. So this I have already told you how to proceed for a new thing. What is the significance of this poly thing? You see that these are this is a kind of Pissolithis mushroom ectomycorrhiza to the eucalyptus tree. You can see that when ITS regions phylogeny was done, it was found that all this grown on whether it is from Australia or Brazil, all the Pissolithis that was found in eucalyptus collided differently. All the thing that was Brazil, Kenya, and even this thing collided differently. Although Kenya eucalyptus collided, that means its origin eucalyptus. If you are following morphology, then you have said that this Kenya eucalyptus have originated from the Australia and this eucalyptus, because you are following morphology. But the moment you did phylogenetic study, it was colliding here differently, because it has origin from here, not from here. Okay, so this is the kind of resolution, this is the kind of help that it is giving. For morphological or identification, you can only find that, okay, these two are different species. But we are doing phylogeny, you can also find that which evolved earlier, which evolved later. Okay. So for if you are doing whole genome, so you have to calculate AINI calculations. So now good pipelines are available, like Fungi DB is there, then Microcosm is there. So all these pipelines are already available on the net. You can search. You can just have to give your whole genome data, upload it. It will take at least 5-6 hours or overnight, and then you can calculate the ANI based on those kinds of things. But it's still not mandatory for our thing. So I'll skip and Major changes in the code of nomenclature. So these are the things, that is no Latin description required, one fungus, one name. Registration of your name in Microbank is compulsory now, and definitely electronic publication is okay. And one fungus has one name, no two names have to be given. So these are some of the data. FungiDB is good for every. I follow still, and has two things. One is type things also, type sequence you can compare it with, just click it. One is non-type, so follow still the GenBank because that is having. Those who are working on LSU and the whole genome, then information you will get all these. The taxon is still not started very nicely for the guy, but also now for whole genome FungiDB is kind of good publications that is published by studies in ecology, where whole genus has been now, and follow this, are freely available on the website, published by Netherlands. Kind of manuals like by Fusarium and Coma or Alternaria, these you should have in your labs for the morphological that thing. Without that, you. Why we are following the polyphasics? Because you know that we have, it's our 99% of our genome is similar to them, but still we are quite different. I hope we have 99% similar to this, but still I look quite different from. Yes. So the review article that we publish, that when you morphology able to have some ambiguity, the more you add technology, then you will be able to resolve the. So our taxonomy by the technology. At one time we had the technology, we followed that. Then it came SEM. Then we were able to solve based on the SEMs. One having smooth spores, one having spotted spores. Then we followed the sequencing. Then we are following the whole genome sequencing. We are dependent, thanks to the biophysics persons who are working harder and harder to. There are two you can follow, but this is. But now, this we have not covered the whole genome, but now the whole genome. Some things that I'll out about the technologies that are also available for diagnosis. These are not for taxonomy, for diagnosis. There is a difference between diagnosis and taxonomy. Malditoff, you just tap your culture, treat it with formic acid, nitric acid, it will extract all the protein. 1 hour, it will has around more than 4,000 database of fungi are there, so it is good diagnosis because it is very fast. So one way you can also have, you will have you can add your own database also if you're having new species. This is Raman spectroscopy, very nice, very costly, around 2 crore, but very nice because it less specimen all else. Although this I want to that basic research and applied research are two different processes. One must have not expected this, but now we are. Why it is nice? Because it does not use, when you have a less specimen, it does not, you can pipette it out also your specimen. It is non-consumable. Whole genome website, you can follow it, are being published. Lot of Either you will not understand anything, or I'll ask madam that there should be a questionnaire at the end of the, then the certificate will be given. Yes, yes. Then I'll receive many of the mails, sir, please forward the presentation. Yeah, you will either, first of all, you have to first extract the compound that you are doing it, extraction by either ethyl acetate or whatever compound for this, then try to extract it, then you purify and run into the HPLC, then you will see. Yeah, based on the literature, if you see that you have found, suppose Aspergillus, Fusarium, Coriatrichum, these are the complex genera. 5 genera are only Aspergillus, Penicillium, Coriatrichum. These are the 4-5 genera. Found any of this, and you have done the morphology, you have done the all the physiological things, you have done ITS sequencing, you have done LSA, MLSA based on the that have been based. Still, you are finding that your both the species are coming in the same clade. They are not separating as the two branches. If you have a two branches, that means these are two different species. This is very clearly resolved by MLSA. But you are finding that these are not two different branches, or the confidence limit of that two branches are not very good. Very small branch is given looking into that. So at that stage, you will try to do a extralite profiling of those two strains, and you will compare it with the, not with the all, only with the second species that is coming into the same thing. Because you others is already resolved by MLS. Only those two, one only you are not able to. You will compare your extralite with that, where the complete branch is not there or a small branch is there. Here branch is there in your phylogeny, then that means there are two different. I'll just add like what she has said. You'll be learning phylogeny in the have there. Try to interpret the phylogeny. Phylogeny tree you will be able to learn. Try to have the interpretation of the phylogeny. How to interpret it? Monophylogenate, polyphyletic, why it is polyphyletic, whether my strain is polyphyletic, at what places my strain will be a new species clearly and where it is not clear, and what is vertical distance and horizontal distance, because these things will come into mind when you
study, but I very not I heavy reference. Thank you. Just to victim.